All right, let's jump right into this explainer. We're looking at the Great Space Paradox, unpacking the prokaryotic genome. We usually think of biology strictly in terms of chemistry, right? But today, we're tackling a pure, mind-boggling mystery of physical space and structure, specifically how a microscopic bacterial cell organizes its massive genetic blueprint. Just pause and think about this scale here for a second. How do you fit 2 entire millimeters of DNA into a cell that is literally 1,000 times smaller? We're talking 1 to 2 micrometers in length. It's the ultimate biological packing challenge. It's kind of like trying to cram a mile of insanely tangled headphone wire into a tiny matchbox. Oh, and you still have to be able to pull out exactly the one piece of wire you need, the exact second you need it, without tangling the whole shebang. Notice the sheer physical impossibility of that task right here. On the left, if you just let an E. coli genome float around freely, its natural polymeric properties make it puff out into this massive tangled ball. We're talking a volume of over 500 cubic micrometers. But in vivo, meaning inside the actual living cell, it transitions into this beautifully organized, highly condensed structure that takes up less than a single cubic micrometer. It's wild. So to pull this off, the bacteria has to achieve a mind-bending 1,000-fold condensation of its genetic material. 1,000-fold! It literally forces that massive random coil down into a tiny fraction of its original volume. And the crazy part? This extreme compaction process is happening every second of every day in literally every bacterial cell on the planet. Now, this tightly packed little marvel has a specific name, the nucleoid. It's defined as the densely packed region of a prokaryotic cell containing the DNA organized purely by architectural proteins. But here's the real kicker. Unlike our own cells, which lock DNA away inside a rigid membrane-bound nucleus, the bacterial nucleoid is completely membrane-free. There are 0 physical walls keeping this dense genetic downtown district contained. It's purely a masterpiece of structural engineering and physical forces holding the whole thing together.
And man, this brilliantly shows what happens when you rupture that invisible structure. Check out this electron micrograph of an exploded bacterial cell. Do you see that huge radial explosion of tightly wound DNA just spilling out everywhere? I mean, it perfectly proves the intense coiled physical pressure that's normally trapped inside that nucleoid. The cell is literally bursting at the seams with genetic info.
So the big question is, how do they actually pull off this compaction without a rigid container? Well, the first step is a mechanical solution, driven by an enzyme called DNA gyrase. It creates negative supercoiling. Think of it like taking a rubber band and twisting it between your fingers over and over again until it starts coiling up on itself. That's exactly what DNA gyrase is doing. It forcefully twists and winds the DNA strands into these tight, negative supercoils. This dramatically shrinks the overall length, but it also leaves the DNA primed and ready to unwind easily later for replication. Pretty clever, right?
But, twisting alone just isn't enough. To actually build this structure, the cell relies on nucleoid-associated proteins, or NAPs. If supercoiling is the twisting of the rubber band, NAPs are the clips and ties holding it all together in a very specific shape. They're basically the architectural scaffolding, physically bending, bridging, wrapping, and just bunching the DNA strands into submission. As you can see, each of these structural NPs has a highly specific maneuver for folding the genome into tight loops. Take HAU, for example. It specializes in bending, bunching, and stiffening the DNA. Then you've got IHF, which causes sharp bending. HNS is fantastic at stiffening and bridging 2 different strands together. And FIS tackles both bending and bridging. It's basically a complete microscopic construction crew, where every protein has a unique job to organize the chaos. And the end result? It's this beautifully constrained, functional shape. You can see how the NAPs clamp the twisted DNA into manageable loops, which we call topological domains and macrodomains. Notice how the proteins cluster right at the center, creating this incredibly dense core with loops of DNA radiating outward. Because remember, this isn't just about saving space, it's about organizing the genetic files so they are immediately accessible the second the cell needs them.
Which brings us to Act 2, the efficiency imperative.
Okay, so now that our immense genome is packed securely into this tiny biological suitcase, how does the cell efficiently unpack and read it? Well, the answer is that prokaryotic genomes are just ruthlessly efficient. I mean, a staggering 85 to 90% of their DNA codes directly for functional sequences. Contrast that with us humans, where a massive chunk of our DNA is non-coding. But in bacteria, there is virtually 0 wasted space, almost no introns. Literally every micrometer of that tightly coiled nucleoid is dedicated directly to survival.
To maintain that kind of high-speed efficiency, bacteria organize their genes into these neat little industrial zones called operons. An operon is basically a specialized cluster of functionally related genes that are all controlled by a single promoter to really optimize transcription. It's the cellular equivalent of putting all the machinery you need to build a specific product on one single assembly line, all controlled by one master switch.
Let's look at the ultimate case study in bacterial logic to see this in action, the lac operon. Looking at the structural layout here, right up front, you've got the promoter and the operator regions. Those are your master control switches. And then, right behind them, lined up in a perfect row, are the coding genes, LAC-Z, LAC-Y, and LAC-A. The cell needs all three of these genes to work together to break down lactose for energy.
This operates on such a brilliant step-by-step conditional logic. So, step 1, when there's no lactose around, a repressor protein just sits right there on the operator, completely blocking the RNA polymerase from moving forward. The assembly line is totally shut down. But step 2, when environmental lactose finally shows up, it acts like a key, binding to the repressor and pulling it off. Which leads to step 3. The path is totally clear and the polymerase just zooms down the line transcribing LAC-Z, LAC-Y, and LAC-A all simultaneously. The cell saves a ton of energy by keeping that lactose-digesting machinery locked down until the exact second lactose actually appears.
And because of this brilliant operon structure, bacteria can produce what we call polycistronic mRNA. Eukaryotes, like us, usually produce monocistronic mRNA, where one transcript codes for just one single protein. But prokaryotes? They use this polycistronic mRNA to manufacture a complete set of related tools from one single continuous set of instructions. Seriously, it is absolute peak manufacturing efficiency.
And that leads us to our last section, the final twist, genome plasticity.
Now, despite all of this perfectly packed, highly structured order we've been talking about, the bacterial genome is definitely not a closed system. Actually, bacteria are constantly swapping genetic packages through horizontal gene transfer. And they have three primary ways of doing this. There's transformation, where a bacterium simply absorbs naked DNA just lying around in the environment. There's transduction, where a bacteriophage virus accidentally transfers genes from one cell to another. And finally, conjugation, which is the direct transfer of plasmids between cells through physical contact. By using these methods, bacteria are constantly altering their beautifully packed genomes on the fly, instantly picking up complex new traits like antibiotic resistance.
Which honestly leads us to a pretty fascinating philosophical problem. If genes are just constantly moving horizontally between these brilliantly packed organisms, mixing and matching their core machinery all the time, can we really even define a bacterial species at all? So as you digest all this incredible cellular architecture today, from those twisted supercoils to the brilliant logic of the operon, I'm going to leave you with that provocative thought. It's something that continues to puzzle evolutionary biologists to this day. Thanks for joining me for this explainer.